Hi, good night, everyone. I'm Dr. Randall, and on behalf of the Neurosurgery Department at Norfolk, we'll be continuing the set of lectures on the basics of neurosurgery. So for the second presentation, we're going to be doing the embryology of the central nervous system. So the learning objectives of this class is to, one, outline the key events from fertilization to neural tube formation, so from zygote formation to neurulation. Explain the critical roles of the notochord and the neural crest cells, which orchestrate the patterning and migration that shape our nervous system. Identify the primary and secondary brain vesicles and what structures they give rise to. We're just touching on the development of the spinal cord because that really needs its own lecture separately. But we'll also be talking about the ventricular system and how the ventricles emerge from the neural tube. And then also looking at key timing milestones to correlate them with common neural tube defects so we can know when and how things could possibly go wrong. So before we dive further, I just want to establish the difference between the central nervous system and the peripheral nervous system. So the central nervous system comprises of your brain and your spinal cord, and these are protected by your cranium and your vertebral column. They're basically your command and integration centers of the body. Separately, you have your peripheral nervous system, which includes all the neurons and nerves outside the CNS, and that means your cranial nerves, your spinal nerves, and their associated ganglia. They act as a communication network to link the brain and the spinal cord to the rest of the body. So in the slides that follow, you'll see that both systems actually originate from the same structure, your neural tube, but they diverge based on the differentiation of neural crest cells and neural epithelial cells. So just starting from the very beginning, a light touch on, once fertilization occurs, you're going to have the fusion of your maternal and paternal co-nuclei. That's going to give rise to your zygote, which is going to have rapid mitotic divisions. And by the end of the first week, or on day 6, 7, you're going to have the formation of your blastocyst. So your blastocyst has your outer trophoblast that becomes your placenta, and then you have your inner cell mass, the embryoblast, which will then give rise to the embryo itself. And at the end of the first week, it implants superficially into the endometrium itself. So after the first week, on the second week now, your inner cell mass, or your embryoblast, is now going to divide into your epiblast and your hypoblast. And that's going to form your bilaminar germ disc. Above the epiblast, the amniotic cavity develops, and then below the hypoblast, the yolk sac is going to form. And around this, the extra embryonic mesoderm and the chorionic cavity appear, and that's going to suspend the embryo by the connecting stalk, which will become the future umbilical cord. So week 2 sets up the stage for gastrulation in week 3. So week 3 now, the embryo undergoes gastrulation, which is essentially the process by which your 2-layered bilaminar disc is going to become your trilaminar disc, forming into your 3 primary germ layers, your ectoderm, mesoderm, and your endoderm, which is the foundation for every tissue and organ in the body. So you can see that outlined here, your ectoderm, your mesoderm, and then your definitive endoderm on this diagram. It all begins with the appearance of your primitive streak along the caudal midline of the epiblast. This structure establishes the body's axis, so it's going to give you a cephalocaudal orientation and your true head to tail orientation. Cells from the epiblast is going to migrate inward through the primitive streak. These first wave of cells will displace the hypoblast and form your definitive endoderm. The next layer of migrating cells will spread out between the endoderm and the remaining epiblast, and this is going to become your intraembryonic mesoderm. And the cells that remain will differentiate into your ectoderm. And by the end of this process, you have your trilaminar disc. So after that now, after the trilaminar disc has formed, the next key structure to appear will be your notochordal process, which begins early in week 3. So from the primitive node at the cranial end of the primitive streak, specialized mesenchymal cells will migrate forward. These cells will form a midline rod-like structure called the notochordal process, which extends cranially between the ectoderm and the endoderm. The central cavity of the primitive pits is going to continue into this process, and that's going to create your notochord canal. Around day 17, the floor of the notochord process will fuse with the underlying endoderm, briefly connecting the canal to the yolk sac below. So by day 19, the fused floor is going to degenerate, and that's going to form a temporary opening between the yolk sac and the notochordal canal. In figure 4.6, if you can see that, you see diagram sketches of the dorsal views of the embryonic disc, showing how it's going to lengthen and change shape during the 3rd week. So the primitive streak lengthens, again, by the addition of cells at its caudal end, and the notochordal process lengthens by migration of cells from the primitive node itself. Moving on to neurulation, now that the notochord is established, it's going to begin its most critical function, which is triggering the formation of the nervous system. And this process is called primary neurulation. So during gastrulation, the notochord is going to reach key signaling molecules, most notably sonic hedgehog and BMP inhibitors, which is going to act on the overlying dorsal ectoderm here. And that's in response to that, the ectoderm cells are going to elongate and become columnar, forming a specialized region known as the neuroectoderm. This layer is going to thicken progressively to form a neural plate, which becomes visible on the embryo's dorsal surface by about day 18. This plate is essentially the blueprint of the brain and the spinal cord, and is the first visible sign of the central nervous system taking shape. So continuing on neurulation, once the neural plate has formed, the next step is it folding. So around day 20, the neural plate is going to begin to invaginate along the midline, and that's going to create a central neural groove. And then on either side of the groove, raised edges called neural folds begin to rise and curve towards each other. As this continues, the folds are going to gradually move closer together, and eventually they will fuse in the midline, converting the open plate into a closed neural tube, typically by the end of week 3. The neural tube will later differentiate into your brain and spinal cord again. At the same time, some of the neuroectodermal cells at the lateral borders of the fold separate, and these are going to become the neural crest cells. And these migrate extensively throughout the embryo to form the peripheral nervous system, as well as other specialized structures like the medulla and facial cartilage. So closure of the neural tube now. So it doesn't close all at once. Closure actually begins in the middle of the embryo, and then it proceeds in both directions, cranially and caudally. The process starts around day 21, and it's a bit like a zipper closing in both ends at the same time, with the head end or the cephalic end closing first. By day 25, the cranial neuropore will close, and 2 days later, around day 27, the caudal neuropore closes. If there's any, these closure points are quite critical. So if there's any failure or issues in closure of the neural tube, you can get neural tube defects at this point in time, which we'll discuss later, so the anencephaly when the cranial end doesn't close, or spina bifida when the caudal end doesn't close or remains open. So I just wanted to play a short video on neurulation animation. I hope it plays, just to give a better visual representation of what's taking place. So I'll just play that there. Okay, so I don't think this video is filming. Yeah, well, I'll forward the video to the group later after the presentation. But essentially, it will just show the folding over the closure of the neural plate, essentially, to form a neural tube. So just a quick touch on the basic organization of the spinal cord. So after closure, the neural tube will differentiate into distinct regions. You have the ventral part and your dorsal part. The ventral part will give you basal plate, which will give rise to motor neurons, and your dorsal plate, which will give you alar plate that forms your sensory neurons. And these are separated by the sulcus and the midlands, and that's the basic organization of the spinal cord. But again, that will be a separate lecture. Moving on to the further development of the brain itself, you're going to have your early brain structure. So by the beginning of the 4th week, the neural tube was fully closed, and we start to see regional specialization, particularly at the cranial end. So the rostral end of the neural tube expands and balloons out into 3 distinct enlargements, known as your primary brain vesicles. The most anterior is a prosencephalon, or your forebrain. Behind that, your mesencephalon, or your midbrain. And finally, your rhomencephalon, or your hindbrain. Each of these will further subdivide as the brain becomes more complex as it continues to develop. Around week 5, these 3 vesicles will differentiate into 5 secondary vesicles, which will give rise to your major adult brain structures. So this diagram here shows a pretty well representation of it. You can see the transformation we just discussed, which is your primary brain vesicles going to your secondary brain vesicles, and then finally your adult brain derivatives. So just starting at the top, you can see your prosencephalon, or your forebrain dividing into telencephalon and your diencephalon. The telencephalon will give rise to your cerebral hemispheres, while the diencephalon will form your deep midline structures such as the thalamus, the epithalamus, hypothalamus, and pit ventricle. The mesencephalon or your midbrain remains undivided, but it develops into the midbrain structures, including a tectum and tegmentum, and that contains the cerebral aqueduct. And then finally, you have your rhombencephalon or your hindbrain, which is going to give your mesencephalon and your myelencephalon. Your mesencephalon will form your pons and your cerebellum, and the myelencephalon will develop into the medulla oblongata and the lower portion of the fourth ventricle. So as the embryonic brain continues to enlarge, it doesn't just grow in size, it's also going to bend eventually. So that's going to create your first major curvatures of brain fractures. So initially, you're going to have two primary bends appearing. The first is your midbrain fracture, which develops in your mesencephalic region. The second is the cervical fracture, which is located between the hindbrain and the spinal cord. These bends help the development brain accommodate its rapid growth within a limited space, and as the development continues, unequal expansions between these regions lead to the formation of a third opposite bend, the pontine flexure, which curves dorsally. The pontine flexure will play a crucial role in causes spinning of the hindbrain roof, which shapes the fourth ventricle and sets the groundwork for the future pons and cerebellum. So moving on to individually each of the secondary brain vesicles, the telencephalon is the most rostral part of the brain vesicles and ultimately gives rise to the cerebral hemispheres. Before we look at the growth, again, it's important to recall the concepts of neural plates. The early neural tree was organized into two functional regions, your basal plate, which will form your motor structures, and your alar plate, which will give rise to your sensory and higher order association areas. In the forebrain, the alar plate becomes dominant, driving the massive expansion of cortical tissue. Structurally, it consists of two large lateral outpouchings, which form the cerebral hemispheres again, and the median portion, which forms the lamina terminalis. As development proceeds, the hemispheres expand laterally, anteriorly, and superiorly, led by the rapidly growing neocortex, which will account for about 90% of the cerebral surface. The outward growth of the alar plates causes the hemispheres to overgrow neighboring regions, such as the diencephalon, the midbrain, and the hindbrain. That's going to produce the complex overlap we see in the mature brain itself. As the hemispheres enlarge and meet in the midline, mesenchyme trapped within the longitudinal fissure condenses to form the falx cerebri, and likewise, mesenchyme also separating the occipital lobes from the cerebellum will give rise to the tentorium cerebelli. This diagram basically just shows or shows how you have your cerebral hemispheres kind of forming over your diencephalon, and you can see it right here, essentially folding down over the fifth ventricle, right, the diencephalon. By about week 6, a prominent swelling called the corpus striatum appears on the floor of each hemisphere. Because the hemispheric floor expands slower than its thin cortical walls, each hemisphere is going to adopt a C-shaped configuration, which also defines the curve of the lateral ventricles. The caudal end then bends ventrally and rostrally to form the temporal lobes. As cortical differentiation continues, white matter fibers develop that split the corpus striatum into two components, giving you your caudate nucleus and your lentiform nucleus, made up of the pitumen and the corpus pallidus. These fibers will unite as the internal capsule, another C-shaped structure that mirrors the curvature of the lateral ventricle. Moving on to the diencephalon, this develops from the median portion of the prosencephalon, and it's organized into a roof plate, two alar plates, and is enclosed by the third ventricle. Its boundaries are set posteriorly by the pineal gland and mammary bodies, and anteriorly by a plane just rostral to the optic chiasma surrounding the foramen of Monroe. Along the lateral walls of the third ventricle, three swellings will appear in the epithalamus, thalamus, and hypothalamus. You're going to have two grooves mark their separation, the epithalamic sulcus, which divides the epithalamus and thalamus, and the hypothalamic sulcus, which separates the thalamus from the hypothalamus. As the thalami enlarge on both sides, the third ventricle becomes slit-like, and in roughly about 70% of embryos, the thalami fuse across the midline to form the interthalamic adhesion. The hypothalamus forms from neuroblast proliferation ventral to the hypothalamic sulcus, with the mammary bodies emerging on its ventral surface. And the epithalamus develops from the roof and dorsal wall of the diencephalon, and that's going to regress in size relative to the surrounding regions. The pineal body that will arise as a median diverticulum from the caudal roof. By about week 7, continued cell proliferation will convert this diverticulum into a solid gland. So The pituitary gland, or the hypophysis, develops from two distinct embryologic origins that later unite. The first component is the glandular portion, or your adenohypophysis. Around midweek 4, it forms as an upward ectodermal invagination from the roof of the stomatum, known as Rathke's pouch. And Rathke's pouch differentiates into your pars distalis, tuberalis, and intermedia. By about week 6, it's going to lose its connection with the stomatum and become the definitive anterior pituitary. And then secondly, you're going to have your neural portion, or your neurohypophysis, which is actually developed from a downward invagination of the diencephalic floor, derived from neuroectoderm. And this will form your median eminence, your infundobular stem, and your pars nervosa, which make up your posterior pituitary gland. The dual origin basically explains why the pituitary gland is composed of two different tissue types: the adenohypophysis, again, derived from the oral ectoderm, and then the neurohypophysis, derived from the neuroectoderm, which forms the posterior lobe. With respect to sulcal timeline and cortical maturation, by the start of the fetal period, around week 9, the brain surface is completely smooth or lyssencephalic. But over the following months, cortical growth is going to increase rapidly, and it's going to give rise to your sulci and angyri as they begin to form. So this process is what's cortical maturation, and it starts deep in the insula and spreads outward across the hemispheres. And it's driven by three parallel events: neuronal migration, expansion of the cortical gray matter, and increased vascularization to support that metabolic demand. By about between weeks 8 and 10, the first visible landmarks will pair your log into a little fissure, which will divide your two hemispheres. By week 14 to 16, your lateral or sylvian fissure will form, followed closely by your early central sulcus. Weeks 20 to 22, the parietal, occipital, and calcarine sulcus appear, and we start to see early folding of the occipital lobe. Weeks 24 to 26, the primary sulci deepen and the secondary sulci branch from them, which signals a rapid increase in surface area and connectivity that the brain is undergoing. By weeks 28 to 32, the rate of gyration accelerates again dramatically. The insula at this point becomes buried beneath the overgrown frontal, parietal, and temporal lobes. The brain essentially folds in itself within these lobes. And then finally, weeks 36 to 40, the tertiary sulci will emerge, giving the cortex its mature, highly folded appearance. And functionally, this allows a huge cortical surface area within a confined cranial volume. This diagram just shows how the cortical surface will mature over time, showing you week 14, 26, 30, and 36. Going more into the brainstem structure, starting with the mesencephalon. So the mesencephalon, or the midbrain, is the most primitive part of the brainstem, and it actually undergoes the least structural change during the development. It retains the basic neural tube organization with a roof plate dorsally, a floor plate ventrally, and the alarm basal plates on either side separated by the sulcus limitans. As the brain expands around it, the neural canal within the midbrain becomes narrow and elongates, forming the cerebral aqueduct, which connects the third and fourth ventricle. Neuroblasts from the alar plates migrate into the tectum, or the roof of the midbrain, and differentiate into four prominent nuclei: the superior and the inferior folliculi. These nuclei serve functions. The superior folliculi mediates visual reflexes such as tracking and eye-head coordination, while the inferior are responsible for auditory reflexes and sound localization. So within the segmentum of the midbrain, neuroblasts derived from the basal plates give rise to several key neuronal groups. These include the red nuclei involved in motor coordination, cranial nerves 3 and 4, which are responsible for your ocular movements, and components of the reticular formation that help regulate arousal and autonomic activity. Adjacent to the ventral surface, the substantia nigra begins to form besides the two cerebri, or the cerebropeduncles, and its exact embryonic origin is somewhat debated. Some evidence suggests that the near is from the migrating cells of either the alar or basal plates. The descending fibers from the cerebrum extends through the ventral midbrain to form the two cerebri, which carries three major tracts: your corticopontine, corticobulbar, and the corticospinal. And as the midbrain matures, these peduncles enlarge and become more defined, forming the conduit between the forebrain, hindbrain, and the spinal cord. This diagram here just shows the mesencephalon development. The A is the end of the 5th week here. B shows a transverse section of developing midbrain, showing the early migration of the cells. from the basal and the alar plates. See here, you can see a sketch of the developing brain at 11 weeks. And then DNA will show transverse sections of the developing midbrain at the levels of the inferior and superior colliculi, respectively. The metencephalon represents the rostral portion of the hindbrain, or the raw metencephalon, and it gives rise to two major structures, the pons ventrally and the cerebellum dorsally. The central cavity within this region expands to form the upper part of the fourth ventricle. The development of the pontine flexure plays a key role here. It widens the ventricle by pushing the lateral wall outward and thinning the roof plates, giving the fourth ventricle its diamond-shaped configuration. Within the neural tube, neuroblasts of the basal plate differentiate into motor nuclei, while those from the alar plates form the sensory nuclei, and then this establishes the classic medial to lateral organization of the brainstem, motor medial and sensory lateral. So within the developing pons, the derivatives of the basal and alar plates become distinct cranial nerve nuclei, reflecting the same medial motor and lateral sensory pattern seen throughout the brainstem again. These include the abducens nerve, cranial nerve 6, which controls the lateral rectus muscle, the trigeminal facial motor nuclei, cranial nerves 5 and 7, which innervate muscles derived from the pharyngeal arches and the suprasalivatory and lacrimal nuclei, both parts of cranial nerve 7, which provides the parasympathetic output to the salivary and lacrimal glands. From the alar plate, we see the formation of the sensory nuclei, the principal and spinal trigeminal nuclei, also associated with cranial nerve 5, which processes the facial touch and pain sensation, while the vestibular and cochlear nuclei of cranial nerve 8 are responsible for balance and hearing. And together, these nuclei integrate the core motor and sensory functions of the developing pons. The metencephalon, the cerebellum now develops from the dorsal alar plate thickenings at the rostral end of the metencephalon. Initially, these paired cerebellar swellings, project into the roof of the fourth ventricle, but as they enlarge, they fuse across the midline to form the vermis and the two cerebellar hemispheres. With continued growth, the cerebellum expands posteriorly and superiorly, which will eventually overgrow the pons and medulla. This process is actually guided by, in part, by the transcription factor 6, which directs proliferation and patterning of cerebellar neurons. Migrating neuroblasts from these regions differentiate into two major components: the cerebellar cortex, the deep cerebellar nuclei, particularly the dentate nucleus, and the pontine nuclei, which relay input from the cerebral cortex to the cerebellum. From an evolutionary perspective and functional perspective, the cerebellum can be divided into three main regions: your archaea cerebellum, or your floccular nodular lobe, which mediates the balance and vestibular control; your piemial cerebellum, including your vermis and anterior lobe, which regulates posture and muscle tone; and your neocerebellum, or your posterior lobe, which coordinates scale voluntary movements. Now moving on to the myencephalon. The myencephalon forms the most caudal division of the brain, extending from the first spinal nerve up to the pontine flexure, and it will give rise to the medulla oblongata. As development progresses, the lateral wall of this region rotates outwards around an imaginary longitudinal axis to the floor plate, and this rotation will cause the roof plate to stretch and thin into a single layer of cells, which forms the roof of the fourth ventricle. The caudal portion of the myencephalon closely resembles the spinal cord in both its structure and development. Within this region, neuroblasts derived from the alar plate migrate into your marginal zone, where they form isolated clusters of gray matter. The gracile nuclei medially and the pinnate nuclei laterally. On the ventral surface, the medulla develops a pair of longitudinal fiber bundles known as the pyramids, and these contain the corticospinal tracts that will descend from the developing cerebral cortex. In the rostral portion, as the walls of the medulla move laterally, the alar plates shift to lie lateral to the basal plates, and this reorganization will result in the motor nuclei being positioned medially and sensory lateral. In the developing myencephalon, the basal and alar plates give rise to characteristic columnar arrangement of motor and sensory nuclei that define the adult medulla. So again, you have your general somatic efferent column, lies closest to the midline, and gives rise to the hypoglossal nucleus, which supplies the intrinsic and extrinsic muscles of the tongue. Lateral to that is the special visceral efferent column, whose neurons innervate muscles derived from the pharyngeal arches. These form the nucleus ambiguus, which contributes fibers to the glossopharyngeal vagus, and the cranial portion of the accessory nerve. And further laterally, the general visceral efferent column develops into parasympathetic nuclei, the dorsal motor nucleus of the vagus, and the inferior salivatory nucleus of the glossopharyngeal nerve. Collectively, these motor nuclei are positioned medial to the sulcus limitans in the floor and the ventrolateral walls of the fourth ventricle. On the other hand, neuroblasts from the alar plate form your sensory nuclei, which organize into four columns arranged from medial to lateral. One, you have your general visceral afferent column, which receives sensory input from the thoracic and abdominal viscera. Next is the special visceral afferent column, which carries taste fibers from tongue and epiglottis. Then you have your general somatic afferent column, which handles tactile and pain sensations from the head and neck, form part of the spinal and principal trigeminal nuclei. And finally, you have your special somatic afferent column, which receives input from the vestibular and cochlear systems, forming here the vestibular and cochlear nuclei of cranial nerve beads. So just a touch on ventricular development. The diagrams here show development in ventricles at day 36 compared to 3 months in. So at day 36, you can see that the brain is still basically a series of hollow vesicles. You can see your prosencephalon, your mesencephalon, and your rhombencephalon, each with a central cavity. Those cavities, which will eventually become your lateral third and fourth ventricles. You can see that the cavities are still relatively straight and continuous, and there's minimal bending at this point because the brain hasn't developed this full flexures yet. By 3 months in now, it's a different story. The rapid expansion of the telencephalon hemisphere stretches and clears the lateral ventricles with the characteristic C shape that you see, following the same contour as the growing cerebrum. The diencephalic cavity remains as the third ventricle now, narrowed by the bulging thalami on either side, and the mesencephalic cavity has elongated to form a cerebral aqueduct, connecting the third and fourth ventricles. Down the hindbrain, you can see the rhombencephalic cavity that has flattened out and widened because of the pontine flexure, creating the fourth ventricle and its diamond shape. The choroid plexus begins developing very early, right after neural tube closure at 4 distinct sites along the roof of the neural tube. Around day 9.5, the epithelium begins forming, and that peaks by about day 11 to 12, and that's going to complete by day 14. Structurally, the thin ependymal roof of the ventricles become covered externally by pia mater, which is self-originates from mesenchymal tissue, and together these layers will form a telechoroidea, which is a vascular membrane that lines the ventricular roof. With the active proliferation of the pia mater, the telechoroidea will start to invaginate into the fourth ventricle, and these folds will later differentiate into the choroid plexus. You're going to have similar processes occurring in the roof of the third ventricle and the medial wall of the lateral ventricles. The epithelial component of the plexus is derived from neuroepithelium, while the stroma comes from mesenchymal cells. Functionally, the choroid plexus secretes cerebrospinal fluid, which is later modified by the brain spinal cord and meninges, and it also contains key signaling molecules like retinoic acid, which influence neurodevelopment. As the roof of the fourth ventricle continues to develop, it will evaginate to form the median and lateral apertures, or the foramina of Magendie and Luschka, which allows CSF to flow into the subarachnoid space. And then finally, absorption of CSF occurs via arachnoid villi, which project into the dural venous sinuses to complete the CSF circulation pathway. This diagram here just shows a transverse section to the diencephalon and the cerebral vesicles at around day 50, of 1 or 50 days. What we're seeing here is we're looking at the level of the interventricular foramina, where you can see the lateral ventricles of the cerebral hemispheres communicate with the third ventricle of your diencephalon. You can see that the thin medial wall of each lateral ventricle here and its junction with the roof of the diencephalon lies the choroid fissure itself. That fissure will mark the point where the choroid plexus begins to invaginate into the ventricle, essentially where it's going to form the primitive choroid plexus itself. So commissures are the interhemispheric fibrous systems that allow communication between matching cortical areas. Most of them develop through the lamina terminalis, the midline junction between the two telencephalic vesicles. The first to form is the anterior commissure, appearing at the end of the second month. Its anterior fibers connect the olfactory bulbs, while its posterior fibers link the temporal lobes, including the hippocampal gyri, which all pass through the inferior part of the lamina terminalis. By the third month, the hippocampal or fornix commissure develops. Fibers from each hippocampus travel along the posterior pillars of the fornix to converge at the lamina terminalis. Then straightforward as the anterior pillars to reach the mammillary bodies. The corpus callosum, the largest commissure, starts forming around week 10, and it begins as a small bundle just ahead of the hippocampal commissure, but it rapidly expands with growth of the neopallion, arching over the roof of the diencephalon. This forward expansion stretches the lamina terminalis and leads to the formation of the septum pellucidum, and by the 6th month, the corpus callosum is complete. Just below the pineal stalk, the posterior and habenula commissures form, while eventually the optic chiasma develops, where fibers from the medial halves of the retina cross the opposite lateral geniculate bodies. Because of the brain's complex embryology, malformations are relatively common. Most major defects, such as menincephaly and encephalocele, will stem from failed closure of the rostral neurocord during the 4th week. And you have many contributing factors, including genetics, nutrition, particularly folate deficiency, and environmental influences. And these defects may arise from abnormal tissue formation or from disruption in adjacent embryonic structures like the notochord and the cranial mesenchyme. So here, the earliest problems occur with neural tube closure. Around day 25, failure of cranial neurocord closure results in anencephaly, where the calvarium and cerebral hemispheres fail to form, leaving exposed neural tissue, and is usually incompatible with life. When closure is incomplete, but the cranial vault forms, we may see encephalocele or cranium bifidum. Herniation of brain and meninges through a skull defect. Often it could be occipital or anterior or frontal. These patients can survive, but often have neurological deficits, seizures, or hydrocephalus. You have microcephaly, which reflects impaired neuronal proliferation or migration, giving rise to small brain and head size, and causes to range from congenital infections like cytomegalovirus or Zika to teratogens such as alcohol and valproate. Next, in the midline commissural system, you can have a genesis of the corpus callosum when the commissural fibers fail to cross the lamina terminalis. On imaging, you'll see what's considered the classic Batwing or racing car ventricles. Clinically, it can present with developmental delay or epilepsy, although some cases could be asymptomatic. And you can have hydrocephalus, which is essentially an imbalance between CSF production and absorption. You can have different causes of hydrocephalus. You can have, when flow is blocked within the ventricular system, such as primary aqueductal stenosis or PRA type 2 malformation. You can have an obstructive hydrocephalus, or when absorption is impaired, as after a meningitis or periventricular hemorrhage. You can have communicating hydrocephalus. In infants, this manifests as a rapidly enlarging head, bulging fontanelle, and also sunsetting eyes. And finally, you have holoprosencephaly, hydranencephaly, and PRA malformation. So, starting with holoprosencephaly, this occurs when the forebrain fails to divide into 2 hemispheres during weeks 4 to 6. In its most severe form, A-OBA, you get a single ventricle and few hemispheres, often accompanied by striking midline facial anomalies like cyclopia or a single nostril. Milder forms such as semiloba and loba show partial division. Hydranencephaly looks superficially similar on imaging, but is pathologically distinct. Here, the brain forms normally at first, but the cerebral hemispheres are later destroyed, usually due to occlusion of the internal carotid arteries or severe intrauterine infections, such as CMV. What remains will be a thin membrane filled with CSF, with the brainstem and cerebellum still intact. And these infants often present with macrocephaly, positive transillumination, and intact primitive reflexes, but no cortical function. And lastly, we have PRA malformations. So this represents a downward displacement of cerebellar structures to the foramen magnum due to a small or due to a malformed posterior fossa. Type 1 is the mildest, involving descent to the tonsils, and it could often be discovered incidentally or when a syringomyelia develops. Type 2 or your PRA descent to the vulnus, medulla, and 4th ventricle, and it's almost always associated with a myelomeningocele and hydrocephalus. Type 3 is rare, and it involves herniation into the encephalocele. So with that, touching on the fetal abnormalities you can get of the brain, that brings me to the end of the talk on CNS embryology. Thank you.